Hi, I'm Ariel Johnson, a PhD student in Margaret Frank's lab, co-advised by Gaurav Moge, and I would like to tell you about a medically relevant genome with Synteny secrets. Euphorbia peplis is a plant also known as petty spurge or cancer weed. It produces a terpenoid compound, ingenol mebutate, that is used as a pre-cancer treatment. We're interested in this plant for its terpenoids and also for its utility as a model species to study laticifers, which are the cells that produce and contain plant latex. Because the genome was small at under 400 megabases, we decided to do a chromosome-level genome assembly. Our genome is a PacBio HiFi CCS assembly assembled with HiFi ASM and the juicer pipeline, masked with repeat modeler and repeat masker, and annotated with RNA-seq data from 6 different tissues using the breaker pipeline. The pseudochromosomes have an embryo-phyta Busko completeness score of 98.5%. As you can see, our putative centromere is not in the center of chromosome 8, which is incorrect based on observations from chromosome squashes in the literature. That being said, this is an assembly that can be used for chromosome-level Synteny insights. The genus Euphorbia has over 2,000 species and shows massive variation in chromosome number. A.S. Hans in 1973 noted that the base chromosome numbers included 6, 7, 8, 9, and 10, and hypothesized that this variation was caused by aneuploidy, which is where offspring inherit an extra chromosome or a missing chromosome. Hans hypothesized that chromosome duplication caused by aneuploidy was driving speciation in Euphorbia, causing the high species number. However, variation in chromosome number could also be driven by chromosome rearrangement and fragmentation. Now that we have genomic tools, that is a testable hypothesis. Euphorbia lathyrus, which is in the same subgenus as Euphorbia peplis, has a chromosome-level assembly published last year. It has 10 chromosomes in contrast to Euphorbia peplis's 8. This allows us to examine the chromosome rearrangements between the two species. I'm showing a plot made in GeneSpace, a visualization program that runs OrthoFinder and MCScanX for you and shows you Synteny based on gene order. Looking closer, multiple peplis chromosomes align to the same lathyrus chromosomes. For example, the plot on the left shows peplis chromosome 2, and the plot on the right shows peplis chromosome 3, and both contribute to lathyrus chromosome 1. Considering all the evidence, it looks like the variation in chromosome number between these genomes was not caused by simple aneuploidy events, but rather by multiple fragmentation and rearrangement events. I also want to give you an update on the genes that might be contributing to ingenol mebutate biosynthesis. You don't have to understand this whole figure. The takeaway is that the biosynthetic pathway is not entirely known, but Cas9 synthase and multiple cytochrome P450s have been implicated. We located a Cas9 synthase and 2 cytochrome P450s that are the top blast hits from those that have been experimentally validated in other species and showed that they are co-expressed across tissues. We hope to find other putative biosynthetic genes that may be co-expressed with this gene cluster. With that, I'd like to thank my committee, collaborators, and lab mates.